Good afternoon, ladies and gentlemen. Welcome to this breakout session, number 2 of the Anses colloquium on the credibility of scientific expertise. We're having today a webinar that's going to focus on computational and predictive methods for risk assessment. And we will be discussing the promises behind those the methods of so-called in silico or computational toxicology, what it changes to risk assessment, and in particular some of the issues it poses in terms of transparency and what one could call the trust in models and computations on which risk assessment has come over the years to rely more and more. So my name is David Demortain. I'm a social scientist from INRAE here in France, the National Institute for Research on Agriculture, Food and the Environment, and I will be chairing this webinar this afternoon. Again, thank you very much for choosing to attend this webinar. In this brief introduction, I just want to introduce our 4 speakers for today. I will be getting to that in a few minutes, and take a bit of time to preface sort of their presentation and lay out some of the questions that we've asked them to address today. I hope you can see this presentation well. So just to put things in context concerning this use of computational methods, computational models in risk assessment. I want to take a few steps back and talk about expertise very briefly, and just remind this audience of the fact that expertise is not science per se, but it's the translation of science, of scientific knowledge into what can be used as an element of reference for a decision. And we're talking here about difficult decisions made in the context of uncertainty, as we know. And if we follow that definition, then risk assessment, if expertise generally is the translation of scientific knowledge into elements of decision, with risk assessment, we're in the context of a translation of data and a very varied set of data, experimental data, but as you all know, I'm sure there are many other types of data involved in risk assessment. So a translation of that data into a characterization of a risk. For instance, the characterization of a hazardous or dangerous dose. And that is used to, one might say, anchor the decision. So that's the broad context. I just wanted to put that forward, the idea of translating knowledge, translating data, so that that data is useful to make a decision and to pinpoint the risk and make a decision. So moving forward, one simple idea is that there's a lot at stake, a lot happening in that in the span of that translation of knowledge into decision making. And in a sort of very basic way, we know that the people involved in risk assessment select data. They do a lot of work to combine data to make it compatible. They give priority to different studies. They weigh up the value of these different studies to construct the evidence of a risk. They do also a lot of work extrapolating, obviously making calculations to extrapolate between doses, between species, between the animals, animal test and the human. So in that translation, there's a lot of expert judgment involved. There's a lot of interpretive work. There's a lot of human activity involved. And obviously one of the most discussed part of the expert judgment, of human judgment involved, concerns the choice of the so-called safety factors, those numbers that one applies to the results of animal tests to define what's the relevant dose for humans. And although we know that conventional risk assessment has evolved quite a lot beyond that sort of conventional use of safety factors with the human equivalent doses and so on, and benchmark doses and so on, there's still a lot of expert judgment involved in that translation, mediating between the original data and the characterization of the risk and the decision. In many ways, computational toxicology developed, in fact, quite early. We quite often refer to these methods as new methods, but in fact, computational models have been around for a long time, and basically have developed to address some of the limitations of conventional risk assessment. And various methods have been put forward to be able to make risk assessment quicker, not wait for the result of more animal tests, but just make the best of the data that's already available, make it more precise in terms of measuring doses, for instance, inside the human body, make it so less dependent on animal models, costly, lengthy animal models, but also less dependent on human judgment, and perhaps also more realistic in terms of the biology and the complexity of situations of exposure. All of that are things that promises that came with the development of various methods, quantitative methods for risk assessment. And so just to mention a few of these forms of computational toxicology, a few of these tools that we've seen emerging over the years. Kinetic modeling has emerged, structure activity modeling, biologically based dose response modeling, also modeling of large sets of in vitro data, such as that undertaken in the context of the Tox21 program in the US. All sorts of developments that have received a massive push with the publication of Toxicity Testing in the 21st Century, a report of the National Research Council in the US in 2007, and the sequels and the follow-ups, including those in Europe. Perhaps the discussion today will emerge from one simple observation, which is that where we had a human, a toxicologist, a risk assessor, a human expert doing the translation between the data and the decision, with those models, with those computational tools and those all those tools to explore and model large data sets, what we have doing the translation, mediating now between the data and the decision, is a sort of material expert, a model, an expert system, an algorithm. And in one sense, all of the transparency requirements that applied to the human expert now sort of come to apply to the model or the system, the algorithm that's here modeling the data to produce that characterization of the risk. And we, in a sense, have moved from the promise of prediction to the issue of transparency, or at least that's up for discussion. How far is now this transparency imperative? How strong is this transparency imperative? To quote a toxicological modeler that I interviewed a few years ago, that made the point quite eloquently. Decision makers want transparency, accessibility, auditability even. They want to see the code, the math, how the model does the thing it does. There is mistrust of the black box. That's again a very strong claim. That's very eloquently made. That's a take on the situation, but it's at least a good lead for the discussion of today, showing that with more models and more computational tools and more IT tools being used in risk assessment, we're potentially placing sort of a black box at the center of the process. So the questions for today revolve around that precisely. How do we and how in practice is transparency being ensured? What kind of practices, what kind of arrangements do we see in place for accessing what's inside the model, what's inside the data, and how the computational models sort of produce, again, characterization of the risk. How do we ensure that models are reliable, not just in the sense of being free of error, but reliable in the sense of the ability of being sort of, trusted? What is happening around those issues? So these are some of the questions that I've asked our four distinguished panelists to think about. So today, each of them will present their experience, present the developments that they have either participated in or used in computation-based kind of risk assessment. So they will take us into a tour of some of these of the use of these computational methods, and then think about those questions I've raised. So to introduce them rapidly, and I will do so in the order that in which they will present. So we're very fortunate and very happy to have Kathryn Guyton with us. So Kate is a senior toxicologist with IARC, the International Agency for Research on Cancer of the WHO, in Lyon, but addressing us today from the US. And Kate has prior affiliations with the Environmental Protection Agency in the US and the National Cancer Institute. After Kate, we will have the pleasure of listening to Jean-Loup Dorne. Jean-Loup is a senior scientific officer with the European Food Safety Authority in Parma, a specialist of kinetic modeling and more generally of new alternative methods and approaches for risk assessment, following all of these developments for EFSA and their application in risk assessment. Frédéric Bois is a senior scientific officer with CERTARA, a company supporting drug development by means of biosimulation. Frédéric is he's head of mechanistic modeling there. He's a biostatistician and biomathematician, if I'm not mistaken. Previously, a research professor at INERIS and the Université Technique de Compiègne. And finally, we have Dr. Anax Oliveira with us, who is head of toxicology at LHASA, a charity developing data systems and expert system for toxicology. So each of them will have about 15 minutes to present. After each presentation, I suggest we take just a couple of clarification questions so that we can move on to the next presentations and keep some sort of the pace. And we will have, after those presentations, ample time to discuss both the take of the different speakers on computational methods, their transparency, and the building of trust in computation. Again, thank you very much for attending. And without further ado, I'd like to give the floor to Kate Guyton. Thank you very much.
Hello, everyone. Kate Guyton, I'm the senior toxicologist in the IARC monographs program, and it's really my pleasure to speak to you today, and I want to thank the organizers for this tremendous opportunity. So David had asked me to reflect a little bit about my experience at US EPA, and I'm just going to pause my video. So I think it's a little out of sync, so I'll just do that in case that helps everybody. So just reflecting on some of my experience at US EPA, and we published this a number of years ago, and what it really shows is the gap between the chemicals that are actually used in commerce, which are shown on the upper part of this slide, compared to the number of notably publicly available reviews. So in the years since we published this, of course, these have increased, but obviously this gap still exists. And this really highlights a number of challenges. Some of them are listed here. First is, if there is no assessment, does this mean there's no hazard and there's no risk? And secondly, of all those agents that haven't been assessed, how do we choose which ones we should assess? And thirdly, which I'm really going to focus on in my talk, is how could we use mechanistic information to really advance risk assessment and bridge this gap? So you can think about what risk assessors have before them is really something in their toolbox. So the decision context and the data availability really drive the selection process for the types of assessments you might do. And these listed along the top of the slide are some of the assessments that are done by US EPA, like the integrated science assessments, which really are done for very few chemicals and rely on human studies, all the way over to high throughput risk assessments, which are really using high throughput data. But this doesn't really get to the issue I presented on the previous slide. Is really, how do you get to everything? How do you get to all in every chemical? And could systems biology data really help us? So to address this challenge, I just want to show that there are some opportunities to do so. And we published this recently, which is a conditional toxicity value predictor. And this predictor actually predicts quantitative risk assessments. It doesn't predict hazard, but it uses, regardless of what's available, whether it's a chemical descriptor, in vivo data, or in vitro data, and uses different types of methods. And the outputs of this are actually quantitative prediction of risk. And this idea is not new. Lauren Zeiss published a paper back in the 1980s noting that you could really use LD50 data, which exist on a whole lot of different chemicals. Toxicologists really don't talk about these data much, but actually they could be used to predict risk. So this is a way to integrate across the different types of inputs that are available to provide those outputs. So to the second question of how do you select and prioritize agents for assessment? And I want to just mention this is just one example that we were faced with recently in the monographs program. We had a number of pesticides that had been nominated for assessment, really thousands. And how are we going to choose? We're not able to do, it's a low throughput process, the IARC monographs, and how could we choose which ones to evaluate? So in order to do that, we developed a comprehensive list of pesticides that are available on the market in the world, and then really automated text mining of public databases. And I just want to emphasize here the importance of public availability. In this visualization here, the size of the node indicates the amount of information available, and the closeness of the nodes indicates chemical similarity. And what this shows you for two classes of pesticides, how much information in the public domain is available, and this really supported as to why we then evaluated some chemicals of this class a few years ago in the monographs program. So in the monographs program, the evidence is integrated in reaching hazard identification conclusions. And this really comes from three disparate lines of evidence: studies of cancer in humans, and these are your traditional epidemiology studies. Now, all of us know these are very large, they're very expensive, and they're actually declining in number. We also have studies of cancer in animals. These are your traditional bioassays. We're all familiar with them. They're also declining in number. And studies on carcinogen mechanisms are actually increasing in number and complexity, and they can be pivotal when the human data are not sufficient. And it's important to recognize that there's diverse sources of mechanistic data. They could come from studies of biomarkers in humans, and we know these are highly human relevant, but they're also costly and complex. And they also range through low throughput, medium throughput, high throughput, and into ultra high throughput. And all of these have different strengths and weaknesses and provide different opportunities. So this all sounds really exciting for mechanistic data, but I just want to point out some of the challenges that were noted when the monographs program in the volume 100 decided to review all of the known human carcinogens. So these are a diverse set of compounds ranging from metals to different types of pharmaceuticals, cigarette smoking. You can think about your favorite group 1 carcinogen, and that was included in this review. And the challenges that were noted is really there was no method to really systematically search for relevant mechanisms. This led to a lack of uniformity across the different assessments. And there's also a different challenge of how do you analyze this database efficiently? And all of these really lead to the last challenge, which is how do you avoid bias towards a favored mechanism? So IARC conducted a couple of workshops on this topic, and it's really credit to Professor Martin Smith, who's at UC Berkeley, who had this idea that we could take these human carcinogens, these are things we know cause cancer, and if we looked at the properties of these, this could be used to really organize the information on mechanisms. And so this is a really new concept. These key characteristics are the chemical and biological properties of established carcinogens. Data on them can provide evidence of carcinogenicity, and for those of us who are involved in systematic reviews, they can be used to assemble data without needing to know upfront what's your hypothesis of the mechanism. So here's the list here. You could think about it as like a top 10 list of what carcinogens do. And I'm going to show you on the next slide just a visualization of this. And really, if you start in the middle here, you can think of something like ethylene oxide. It's electrophilic, it's genotoxic, and it affects these. It has these key properties. But other things like these drugs that are immunosuppressive, this is what they do during their day job, but at nighttime they actually cause cancer by being immunosuppressive. That's actually the key property of them. We have other types of carcinogens that we know, like viruses. Viruses have been in the news a lot lately, but some viruses cause cancer. You can think about the HPVs as an example, and they do this by really causing immortalization and also affecting these other processes and this other key characteristic here, where one cell is turned into many. Now, other types of carcinogens that we know, cigarette smoking, benzene, asbestos, they have many, many of these key characteristics. So this is really the framework that we've been using, and I'm going to talk now about why it's been helpful. And when Martin and I first started to talk about this, I asked him, I said, could we use these key characteristics to design search terms when we're doing our evaluations for each of these, and then we could actually find out if any particular agent up for evaluation has these key properties, and we can organize them, all the information by key characteristic and species, et cetera. And this has really been a tremendous tool for the monographs program as we've applied it. So, and we often get a question of how does this relate to adverse outcome pathways or toxicity pathways? And this is a slide that Martin has provided. It shows a published adverse outcome pathway for benzene and how it causes leukemia, and it seems very plausible. It's metabolically activated, causes genotoxicity, alters cell proliferation, and then a leukemia. But when we actually did a search based on the key characteristics, the picture was more complex. Some of these arrows are not, it's not even clear the direction of the arrows. And there's many other types of key characteristics that are present. So I think that really emphasizes the strength of this method. And we've now applied this in more than 70 different assessments, and this very colorful picture indicates for each of the many different carcinogens we've evaluated that have the different key characteristics. Shows where in which group they've been evaluated, and it shows also the types of evidence, starting from human studies all the way to other types of in vitro and predictive studies where that evidence was found. So, and due to the strength of this method and the experience, when the monographs program updated the preamble to the IARC monographs, which is really our method, in 2019, there was an emphasis on the key characteristics, which we call the KCs, and you can see here that they really can have an impact on the different evaluations that we undertake. And again, this goes to the issue that David emphasized in the beginning. It's because they're linked to human carcinogens. That gives a ground truthing. It gives a confidence. These are sort of the obvious properties of carcinogens, and there's a lot, most experts would actually agree that these are the properties of carcinogens. So this has really been valuable to us as well, and David alluded to this report from the U.S. National Academy of Sciences that did opine upon this approach, noting that it avoids a narrow focus on specific pathways and hypotheses and provides for a broad, holistic consideration of the mechanistic evidence. And the committee also encouraged the development of key characteristics for other types of hazards, and that activity has been going on. On this slide, you can see this very nice picture that we published in Nature Reviews recently on endocrine disrupting compounds. And there's quite a large effort ongoing to develop these key characteristics for other types of toxicants listed here. This effort relies on the expertise of people such as yourself, and if you'd like to get involved, please come to this website and you may be able to engage in one of the ongoing projects. So I'd like to stop there, acknowledging the monographs team, the support that we've had, and I also want to thank you all for your attention. So thank you very much, and back to you, David.
Thank you, Kate. Thank you very much for introducing us to so what I understood is a sort of experiment in also text mining also at the IARC to build a model of carcinogenicity and of mechanisms and to be able to use them more routinely in the assessment of chemicals. It was very enlightening. Sorry, thank you very much. I remind everyone in the audience that you can raise questions through the chat. We can take a few minutes if there are questions that are directly related to Kate's presentation and need some clarification. And I see one from Better Meek, which I'll raise for Kate. Do you think that collaboration to consider key E's or key C's perhaps in a more meaningful integrative construct to additionally inform hazard characterization would be helpful? Kate?
Yeah, so I think that's a great question, and I think one of the things about this approach is about the Casey's approach is it really has relied on a very, very large group of collaborators, and it's certainly the privilege of IARC to bring together world experts on the topics that we consider. So I think that definitely a collaboration is really not only where this has come from, but also where it will go into the future. So thank you for that question.
All right, we have a second question from Annabelle Demy. What are the relations between cases and test guidelines on the OECD side? What's the connection here, if any?
Yes, that's a really great the KCs are really the properties of carcinogens, but each KC, of course, would have endpoints that are linked to it, and really the guidelines produce data on those endpoints. So you can think about, for example, the tests of genotoxicity. All of those types of data would be brought to bear on the question of whether a compound is genotoxic. So it's really, that's your question. Is this genotoxic? Then you bring in those data to really answer that question, yes or no. So I hope that really addresses your question.
Could I add one quick question? I mean, you've presented key characteristics as a sort of end product, finished product. Perhaps it looks deceptively easy to produce them. I mean, I guess you used a very large volume of studies and data to sort of distillate those key characteristics. Can you tell us in practice what it entailed? I mean, this work to derive the KCs.
Yeah, that's a really good question, and I can't really understate how much work it really was. Just coming to the idea of it was really what we would call an aha moment, where Martin Smith came down and said, you know, this is really the direction to go if we could come up with these properties. But if I can go, maybe I can just go back to this list. Actually getting to a consensus on this list of properties, some of them are really obvious. I think we can all, if you just take some of the ones at the top, they're really obvious, right? I think we've known for a long time that carcinogens are genotoxic, but as I was showing you, that doesn't cover the universe. And actually, in the process, this started as a list of 25 properties. Which, as a risk assessor, it's really too many for to be practical. I don't think we would ever get done with our assessments if we really had to go through 25 different things. And when you have more, so it's really the decision of where to collapse and where to expand was really difficult. And I think as we've put them into practice, you have things like this fifth one of inducing oxidative stress. Well, we know this is a property of many carcinogens, but it's also a property of things that aren't carcinogenic. So there's also a lot of interpretation when you come into evidence. If this is the only evidence you have, I don't think you're going to be ready to make a classification on that. Versus if you know something's genotoxic, I think you would have that confidence. Or if it's immunosuppressive, that's such an important property, it's really standalone, whereas some of these others really are working together. So in many ways, it's about really simplifying scientific judgments. And part of that was done thinking back on those group 1 carcinogens, and a lot of evolution has happened as this has been put into practice, and I think will continue to evolve in the future.
Right. Many thanks, Kate. There was a last question, which I'm saving for the general discussion because it's such a good question that will speak to various presentations about access to code versus confidentiality and proprietary nature of data by Marie-Laure Lefebvre. I'm saving that for later. I guess it's time to move on. Thank you again, Kate, and without delaying it further, I will give the floor to Jean-Loup Dorne from EFSA for his presentation.
Hello, everyone. Thank you, David, and thank you for the organizers to invite me and give me the opportunity to present some of the work we do in EFSA regarding computational toxicology tools. So I will try to present briefly, and I might also pause my camera because it seems like some of the audience couldn't connect correctly, so I will just pause the camera. So I will briefly talk about computational toxicology tools and predictive tools at EFSA in the context of the work we're doing towards the implementation of new approach methodologies in chemical risk assessment. So what I'd like to do first is, of course, to highlight an important part of what we do. Of course, risk assessment is part of risk analysis, and actually, this is exactly what we don't want to happen. To be a little more serious, to place EFSA in the EU food safety system, of course, EFSA provides scientific advice and risk communication, so risk assessment and risk communication, of course, in collaboration with many, many stakeholders in response to the food safety legislation. Of course, research driving the risk assessment, the agro-industry, and of course, control and enforcement, which are the results of the risk assessment, consequence into risk management, and of course, the control and enforcement. In practice, EFSA's scope of activity ranges from farm to fork, as we say. So we're dealing, of course, with plant protection products, a range of other regulated chemicals such as food packaging, food additives, feed additives, novel food, vitamins and minerals, but also plant health, genetically modified organisms, animal health and welfare, and biological hazards such as foodborne diseases. In terms of horizontal and cross-cutting science, the scientific committee of EFSA deals with the production of guidance documents that can be applied across these areas. In practice, we often receive a mandate from the European Commission, actually most often in the context of the food safety legislations. It can be the European Parliament, member states, or if we identify an issue that we have to address within EFSA, such as, for example, chemical mixtures or multiple stresses in bees, recent document published, we would actually self-mandate ourselves. So the questions are formulated for problem formulation, where we have the background and the terms of reference, what is the question to answer, which then results in the production of an opinion or scientific opinion, a statement or a conclusion, which of course may have risk management consequences, and in parallel we communicate the outcome to consumers, media, industry and professionals. A key aspect of the new 178, the EU food law, is transparency in risk assessment. A couple of years ago, Professor David Spiegerhalter pointed out an important aspect of transparency, stating that organizations should not aim at increasing trust, rather aim to demonstrate trustworthiness. And key aspect of that is the information we're using should be accessible, intelligible, usable, and accessible. In that context, we're trying more and more often to formalize what we call the weight of evidence, to put all this type of information together in our risk assessments, and of course highlight related uncertainties. Very, very basic slide to just give you a little bit of background. Of course, in chemical risk assessment we have 4 pillars. And often our risk assessment aim to be fit for purpose, that we use tiered approaches depending on data availability, time availability, and resources. So the more data we have and the more time we have, the better the risk assessment, or the more detailed it can be, or the more sophisticated. If we have 1 week to answer a question and we have no data, we might have to rely on either default values or maybe in silico computational tools to assess hazards, for example. So hazard identification, hazard characterization, of course, where we try to identify and quantify the toxic effects to derive what we call reference points or point of departures, to then derive reference values through the use of uncertainty factors, which are safe levels for a particular species. It can be humans, it can be species of ecological relevance or farm animals, for example. The exposure assessment in food safety is basically the combination of the occurrence of a chemical in food commodities and the consumption patterns in a given species. And of course, the confrontation of the hazard dimension and the exposure dimension will give us the risk. So when it comes to hazard tools that we have developed in EFSA, we have an open source database called Open Food Tox, which is an inventory of EFSA's chemical risk assessment since its creation in 2002. We're talking about over 5,000 substances, reflecting out roughly around 2,200 scientific outputs. It is an easy reference for EFSA staff, but also for stakeholders, including member states, agency, and international organization, where we can use this database for crisis. It's of course free and quick and easy access to all EFSA's chemical hazard data. It avoids the duplication of efforts. We can pin down the chemicals we've already assessed. And it supports the international harmonization and data sharing through the use of OECD harmonized templates to design the data model of the database, and of course, easy ways of sharing the data itself. So the content is currently rather simple. We have information about the chemicals with a range of nomenclatures, the CAS numbers, the IUPAC, the SMILES, and so on. Classification of compounds as single substances or group of substances, whether they're mixtures or formulation. The link to the EFSA output itself, whether it's a scientific opinion, a statement, or a conclusions on pesticides specifically, and the basic toxicological information, mostly the output conclusion on the genotoxicity, whether a compound is concluded to be genotoxic or not, mutagenic as well, of course, and the different reference points for human health, animal health, and ecological risk assessment. Finally, the use of uncertainty factors is also referenced, and the reference values themselves for regulated products and contaminants are referenced. You can download the database, and we recently published a review article which describes the database and its future developments, including computational toxicology tools. We have worked recently on a second version of the Open Food Tox database through consultation of a number of stakeholders, including EFSA staff, other member state agencies, and it seemed that the database was a little bit primitive, so we wanted to include new chemical properties, such as physico-chemical properties, in a structured manner using OECD templates, but also the summary of our exposure assessment from historical risk assessments, giving us a baseline for future risk assessment for given substances. Of course, the EFSA outputs, but also toxicological information, including increasingly the need to have structured toxicokinetic data in vitro to pin down the isoforms involved in metabolism, or generally speaking, toxicokinetic parameters, as well as in vivo, and of course, toxicodynamics in vitro, in vivo, to move towards more mechanistic understanding. The future also holds the fact that we ought to connect it to a modeling platform to basically start integrating those new approach methodologies. And if we don't have the in vivo data, we could start using those computational tools to predict a range of chemical properties, including physical properties or bioaccumulation, kinetics, and toxicity. Here, this slide gives you a few examples on the use of open food tox itself to develop QSAR models. A couple of papers were published recently to develop predictive models for NOALs using the rat data. I think we're talking over nearly 2,000 NOALs, and substructure and physical properties to predict those NOAL values for substances for which we don't have the NOALs. Some QSAR models to predict acute toxicity of pesticides in rainbow trout, and more recently, QSAR models to predict acute contact toxicity and classify mode of action in honeybees. A platform is also available for a range of computational models, which is called R4EU, and it's there's a document which explains how to self-register to give you access to all the different tools. We have, for example, a benchmark dose modeling tool, an MDR, microbial resistance tool, Monte Carlo simulation tools, spatial statistics, and so on. So I recommend to have a look. You might find some useful tools there. Then a few years ago, about 5-6 years ago, we went into thinking it would be very nice if we had generic PBPK models to start integrating kinetic information in our risk assessment, but also calculate safe levels on an internal dose basis. So of course, you know that the toxicokinetics is what the body does to the chemical, absorption, distribution, metabolism, excretion, and toxicodynamics is what the chemical does to the body. Ideally, we would like to understand all the biological steps, the key events leading to adversity, and if we can integrate these steps in a quantitative manner, it will improve the precision of our risk assessments. So that is the kind of general purpose. So building this tool is not an easy task. So what we also need to do in parallel is to develop case studies where we can start integrating what we know about metabolism of a compound with variability. And here is an example of potential integration of data on in vitro metabolism of pesticides, but also contaminants such as microcystins, with the in vivo variability we've generated from meta-analysis using the pharmaceutical database. Other examples, development of generic PBPK models for farm animals to try to predict basically concentrations in organs of interest. For example, if we're talking about pigs or chicken, we would be interested to know what are the residues in the muscle or in the liver for human consumption, or in the eggs. And these tools could support that. Also fish. And the idea is that every model that is produced, we should publish the physiological parameters to calibrate the model, as well as the code behind the model. Still, that's not enough for risk assessors to use these models. We need to make an effort to make these platforms user-friendly. So the idea is to build this TK plate platform where you could access the physiological data, or at least select the species from a menu, input your chemical specific parameters, and then have the options to predict a number of kinetic properties such as TK parameters, AUC, half-life, or tissue residues. If you want to do forward dosimetry, which is the prediction of the TK parameters, or if you have biomonitoring data, you have the internal dose and you would like to go back to the exposure. If we want to compare species-specific toxicokinetics for the refinement of uncertainty factors to investigate interspecies differences, human variability, but also ideally toxicokinetic, toxicodynamic modeling. Of course, the reporting of the sensitivity of the model and also its uncertainty is very important. So if we go back a step and try to bring all this evidence together for risk assessment, we currently working from the exposure and compare this exposure, external dose, so to speak, with a reference point which is based on external dose. If we start integrating the dosimetry, we can actually go into a more mechanistic perspective where we would calculate the safe level from the inside of the body, the internal dose. And on that basis, we could really maybe improve the risk assessment practice and think of it more mechanistically. One of the key tools to combine this evidence together, of course, is the weight of evidence. Whether we're using in vivo data or whether we're using computational tools, we can harmonize the reporting of the conclusions of that weight of evidence by summarizing the evidence itself. And it's assembling the evidence, weighing the evidence, and integrating that evidence to answer a particular question. Just brief concluding thoughts. To go back to the basic principles, risk assessment aims to be fit for purpose to answer the question we have received. We're using tiered approaches and weight of evidence approaches. Open Food Tox provide hazard data for all chemicals assessed by EFSA since its creation. And the TK-Plate platform aims to become an open source platform for modeling kinetics and dynamics in species of relevance to the food and feed safety area. We do hope that this platform will be published in the spring or summer 2021 this year. Obviously, we have to carry on developing illustrative case studies for different applications of these tools and implement them in practice. And last but not least, to do so, we have to train, of course, EFSA staff, experts, and the risk assessment community to use these tools in practice. I would like to conclude on some acknowledgements. A very large number of researchers have been involved in this work, including CERTARA, Dr. Billy Hamzal, Nadia Quigno, and Victor Vichek. INERIS in Paris, Céline Brochot, Frédéric Bois, Rémi Baudouin, and Audrey Grech. Radboud University, Leonie Lautz, Adragas, Jan Hendricks. In Rome, the National Food Institute, Emanuela Testai, Franca Buratti, Susanna Vicchi, Laura Turco, Emana Di Consiglio. The University of Utrecht with Nienke Kramer and Emma Castile. Anses in Paris, Kevin Dorne, Camille Béchaud. Open Analytics involved in developing the platform itself, implementation of the algorithms. Hans Bossier, Dr. Varek, Vergut, and Schau. And colleagues in EFSA, Dr. Cortinas Abrahantes, George Cass, Jean Lim, Tobin Robinson, Jen Richardson, and Anastasia Livanu. I'd like to thank the audience for your attention. And back to you, David. Thank you very much.
Thank you very much, Jean-Loup, for this comprehensive presentation. There's couple of questions perhaps about Open Food Tox. There's one coming from the audience. How representative of the data in Open Food Tox, considering the EU member states? And I was wondering myself, going back to a question that someone asked previously, also what does it contain? Can you include in Open Food Tox some proprietary data, or is it data that's only mined from, well, that's only, it's already a lot, but that is mined from publications and so on? So wide is Open Food Tox? So
very briefly, Open Food Tox contains, for now, historical data from EFSA's assessments, which is updated on a yearly basis. So we are talking again about food additives, feed additives, pesticides, contaminants, and so on. And all the data behind EFSA's risk assessment, and it contains the data that EFSA has used to derive safe levels. Of course, we receive dossiers from industry, so from the assessment of EFSA, the data that has been used from the dossiers to make the conclusion and to set the safe level is in the database. In the future, we are thinking of including more data, and that has to be discussed. The fact that EFSA is moving towards using IUCLID to harmonize the data collection with ECHA might provide further opportunities to include more data sets. And at some point, of course, literature data on mechanistic information might be important, as well as including some of the information generated, for example, by our colleagues at the US EPA. I'm not sure if I've answered the question.
Yeah, thank you for those further details. So you've mentioned kinetic models and various other models that can be sort of developed or used based on the TK platform, or if I'm getting that right. Are these models that are already incorporated in the platform, so with algorithms or pre-written lines of codes that you can run, or is it data that you have to extract and to develop your own model aside from the platform?
So currently,
work on that.
So what we've done is we've published the different models in the literature individually, so to transparently report how they've been developed with the physiological data. And on the EFSA Knowledge Junction, as for open food tox, the physiological data are available for download together with an R code. Now, of course, R codes are still a bit esoteric for some risk assessors, so we are now implementing the algorithms into the platform, and basically to run the model you would have to put chemical specific parameters for the chemical of interest you're interested in modeling, and then you will get an automated report with the sensitivity analysis and the results of your simulations. Now the key aspect of that is that we need to write a user-friendly user guide, if I may say so, because that's will help to support the dissemination and the use of those models, and that takes quite some time because we have to bridge the mathematical background together with the biological background and also the risk assessment applications.
Okay, interesting, fascinating. Well, thank you very much, Jean-Loup, again for the presentation and answers. I think it's time to switch to Frédéric Bois' presentation. Frédéric, I'll leave you the floor.
Yes, thank you very much. Here we go. So I don't know if I will. We'll see how the bandwidth is. So thank you, David. Thank you for the organizers of this session for the invitation. I'm very happy that Kate and Jean-Loup gave presentation quite general so that you can have an idea of the big picture on the use of these tools. I will focus on maybe more the most mechanistic aspects and from slightly a different point of view, but not so different. Sorry, I need to click on those buttons. So first, an explanation. Who am I? I'm a pharmacist, in fact, David, and a toxicologist, but I worked on pharmacist turned bad. I worked on mathematical modeling and statistics, but I did that in the last 35 years now, in academia, in public research lab, government agencies. I was part of CALIPA at some point, in the USA and also in France. So I have had quite a large experience, and recently I moved to work for a private company, CERTARA, and where I participate mostly, as you indicated, for the pharmaceutical industry and pharmaceutical regulatory agencies like the EMA, the FDA, Japanese group, putting together this evidence about where does the drug, chemical therapeutic drug, goes into the body. Is it really absorbed? All these questions, which are also relevant to toxicity assessment, but we tend to do them or look at them for efficacy assessment. But it is a very similar question. But at the same time, I am also the developer of software, a free software called MCSIM, which is distributed by the GNU project, and I have done that since my PhD time, and it's still used. Actually, it is used by the US EPA, for example, in PBPK modeling, and it's used by HTTK for software that is developed by EPA. So in a way, I'm a jack of many trades, and I have quite a large experience in this area and from different point of views. Just a disclaimer that I'm speaking in my own capacity, and I'm not speaking in the name of any of my current or past employers. So what do I do? As I indicated, I mostly work with complex models and not so much QSAR. I think that Anax will tell us more about QSAR, those high throughput models. These are more extensive description of models that describe basically the body and the interaction of chemicals with the body. And the kind of question that we can answer is, for example, how much of a chemical is absorbed by the body? And we can do that now with very little animal data or human data from clinical experiments or clinical studies, but we can also do it just on the basis of the molecular formula of the chemical, its physical chemical properties, or also with the help of in vitro data obtained in cells, for example. Other kind of question is, how long does a chemical stay in the body? Are its effect, the effect of a chemical affected by the presence of other chemicals? That is, are they interacting, or are mixtures of chemicals more toxic, or a mixture of drugs less efficient than the drug alone, for example, knowing that many people take, like in the old age, about 8 drugs, therapeutic drugs per day concurrently, or how to extrapolate observation across species, which is something that is very important in toxicology, environmental toxicology, and then also in human toxicology, or for therapeutic development, or how to replace animal experiments with in vitro data. And that has come in the last, I would say, about 20 years, really at the forefront of the question, with the question of replacement of animal experiments, for example. How can we do that? And I will go into this question a bit more, the use of animals. But also, what are the most susceptible individuals? Who in the population is the most susceptible? And these are not always obvious difference. I mean, you can think that maybe the fetus, a pregnant woman, would be more susceptible. Is it or not? But this can be also differences in your, the ability of your liver to metabolize some product and detoxify it. We are not all equipped the same way because of genetic differences or environmental differences to cope with those exposures. So you can see, for example, here a model that we are using currently, and it describes the whole body, how the compound is dispersed by the blood in the various parts of the body, but also inside the individual compartments. We call them compartment parts of the body, call them organs like brain, the kidney, the liver, the lung, how the chemical here finally disperses or accumulate in eventually some types of cells where it can have specific effect. And all that to understand better, really, what is the concentration of the chemical really where it is having an action. Like if you want a drug that to cure, for example, that COVID, for example, you may want it to target the lung in particular, and you don't want it to go especially in the kidney or be lost elsewhere. So how do you ensure that the compound goes to the lung, and can you check that, and at the same time not too much because of toxicity, and at the same time not enough so that you can have an effect. Okay, so these are the keep hitting on the wrong button. Now, you ask in your framework question, David, about the toxicity testing in the 21st century, and Kate also showed us where we are. So, these are elements of my reflection. I would say, yeah, we have slow and unequal progress, but we have progress. But at the same time, we are very far from an algorithmic government and sort of Robocop agency or something like that. We are far from that. I think that most agree now that animals are not very good predictors of humans, okay? And also that animal experiments should be reduced for various reasons, for most ethical reasons. But it's not a uniform progress, and there are differences between, for example, pharmacology and toxicology. We do not have the same amount of data. In toxicology, you don't do experiments in humans, so at all. While in pharmacology, once you have given the drug to patients, you do actually have some data about what it does and where it goes, etc. There are also differences between countries and regulatory frameworks. So it's not the difference in training levels between the different countries, or just the way the programs at the university. What we are talking about here is things you learn when you go to the university, specialized courses, and it depends how they are taught, which faculty teach them, and which professional are seeing them. This is not uniform across the whole world. And so there is also something that I have really noticed. When you're young and you do your PhD, you think you're going to revolutionize research, etc. But there's a very slow diffusion of methods from research to regulation, slower than what you think typically, and this is for some good reason and some bad reason. Typically, adopting an innovation from a regulatory point of view, you may want to be cautious, even conservative, maybe too cautious. And there are also economic arguments against changing regulations, for example. You cannot always do something about that. There's also the fact that different tools are used in different risk assessment contexts. So and those different tools that you've seen are very different in concept. The QSAR model can be quite simple. The models that I showed you before is many equations, and those that Jean-Loup described, which are difficult to check and understand, actually. Also about modeling, I think the progress of modeling depends very much on the progress of basic science and data gathering. It's not independent. We are not inventing model in sort of a vacuum. We need to check them, we need to validate them, we need parameter for them. And we have the case, as Kate mentioned, of omics data, where data pile up, basically, a flood of data. But the basic understanding of what those data mean is lacking, or is at least lagging behind. And we have not talked too much about that because this is still very research oriented, but artificial intelligent analyses are promoted by some to give us prediction on the basis of such data. But it is true that in this case, we are faced with sort of black box prediction. We don't really understand the reasoning under this, because this reasoning is beyond what our brain is able to do. We are not constructed for that. And And so you get some prediction, but without understanding, and that's a problematic. So where is the science policy dialogue? I alluded to that. So well, you have to know that the 21st Century Vision Report was primarily written for US regulatory agencies, and in the US context, it has been diffused beyond, and different agencies have picked it up, but different agencies have different cultures. In general, Kate will tell me if I'm wrong here. They want to regulate fairly and evenly and use the same rules for everybody. And they do not like improvisation, okay, which can be understood. So rules tend to change slowly, and more slowly than research progresses. And unless misdirected, they stick to their purpose. Okay, and typically the purpose of governmental agencies is consumer or environmental protection. Okay, and in this case, they prefer to err on the side of safety. That is, to be maybe too conservative or too protective of health. But who would reproach that? I usually prefer to base decision on sound science. Okay, and considering all the evidence. Okay, but this is very hard, as Kate mentioned and Jean too. And they tend not to like uncertainty. For me, that's a bit of a problem because there is uncertainty always. Or at least they don't like unknown precision, unknown about what is not known, something like this. It's kind of scary when you try to decide. But less uncertainty requires more data. Okay? And more data is expensive. It takes a long time to get. Okay, and if a preliminary, the precautionary principle, and nobody mentioned it yet. I think maybe Kate did it. If precautionary principle is not applied, then no decision will be taken while you're waiting for data. Right? So that's what she meant. There is no evidence to prove that there is no hazard or no risk. Well, that's not true. So what do we do in this case? But people are not always used to think in this face of uncertainty and decide in the face of uncertainty. So we have to be aware of that. And so for about 100 years, toxicology has used animal and fairly basic methods that were many times flawed, actually, had problems. But I wanted to point to the fact that, and that's something also you mentioned, David, animals are models too. Actually, in the scientific literature, you hear about the mouse model, the rat model. It means that they are sort of analogs of humans. And we want mathematical models. We insist that mathematical models, we would like mathematical models to be transparent, clear, have all the code. But you realize that we don't know the code of a rat. We don't know how the rat works. We just see it dying or see it suffer. But we don't really understand how. So why are we more? Why do we want more transparency about mathematical models than about rat, for example? Because we think that the mathematical model, we know it all, and we should be able to understand it completely. That's probably true, but then you realize that this is an advantage of mathematical model and a problem of the rat. And at the same time, David, when you mentioned that we would like we would like the model to be more transparent or be as transparent as experts were before, human experts. Well, I don't think that human experts have ever been transparent. You've never seen what was happening in the brain of an expert, really, in the back of it. So I'm not sure that I think that actually mathematical modeling and everything, putting the mechanism on the table and really putting the hypothesis and making it clear is actually a progress that is very important. And yes, we think that there can be problem with black box models, but again, animals and humans are also black box when they come to thinking. Okay, to a certain point. So I think that understanding how models and humans have work has made progress too, given molecular biology, cellular biology, but this knowledge needs to be integrated. And this is what the problem is. This is all the question of systems biology. After a while, after knowing what many molecules were or many proteins were in the body, how do they work together? How is this thing working together? And you can't really understand it just looking at cells and molecules individually. So you have to put it together. And the best way to do it so far seems to be systems toxicology or systems biology or systems pharmacology, which is trying by modeling to put all this evidence together. Okay. So this is a set of my thinking about that, or various elements for discussion. Okay. But again, I wanted to really stress that understanding mathematical and statistical models is difficult. Okay, and we have to understand that. Okay, but the dialogue is ongoing, and really, for example, the use of those complex physiologically based pharmacokinetic models, the one that I showed you with the many boxes at the start, is picking up, like you see with the years from 2000 to 2020, how the number of publications, that's scientific publications from academia in yellow, okay, has increased, but also from private companies. They are using them, and there's a dialogue because now, sorry, I forgot the last, in orange, regulatory agencies whose mission is maybe not primarily to publish, but they are starting to publish and give opinions about how those models should be used and what should be there. Actually, Jean-Loup published papers, for example, like that. Kate give guidance on how those models should be used, and that's the occasion of a dialogue between the different actors. But you see really that the use of those models is increasing. Okay, and everybody, I think, agrees that full transparency is essential via peer review of the model in the scientific literature. Everybody can check it. Also public workshops, like today, for example, although we are not technical, but, and also implementation documents that can be made available to the regulator. I'll come back a bit later to that. And at the same time that those models are being more and more understood and used, there is more and more funding by public bodies like EPA on the right, or the European Commission funding projects that develop at the same time data to parameterize those models and also further develop those models and make them more accurate, more precise. So there is altogether a global trend in the regulatory acceptance of those models because they are useful, really. And you can think that maybe I don't believe in conspiracy theory. I'm a very basic scientist. They have not been invented to really distort the truth or hide anything. They have been invented to put together all the evidence and make the best decision possible. I'm not saying that they have not been used sometime, but to sort of hide or twist slightly the story. But that is not the norm, and that's sort of easy to see actually when that happens. So something that is important is assuring the quality and reproducibility of the prediction of those models. Okay, so on average, no one wants bad models. Okay, they lead to bad decisions, they lead to bad outcomes. As I mentioned, there may be some bad players, but there will always be. But the question is how to control that. And one way is quality assurance. Okay. And this is well underway, as Jean-Loup mentioned, as, for example, when we work for the pharmaceutical industry, FDA is very strict about quality assurance because in the end, therapeutic drugs given to people are given in high doses. And the secondary effect, you've seen how quickly some drugs can be withdrawn from the market when we see those effects, really trying to avoid that. Now, there is also an issue about reproducibility. Okay, that is, if I have a model, maybe it's a good model, okay, but if I'm the only one able to run it, that doesn't help much, okay, and also everyone agrees that model prediction should be compared to reality for checking verification, validation, okay, but you have to realize that all models are probably wrong to a certain point, like the animal models. But some of them are more accurate, more precise than others for a given use. Okay, so don't think that the models are universal oracles that will predict everything. This is not the case. Okay, so checking verification of the models are usually mandated by all the users, regulatory, the industry using that, or the NGOs actually monitoring and even using some of those models, okay? So on average, the good news is that, for example, this PBPK modeling, on average, the predictor that has been documented in publications, the predictive performance predictions they make are they do not seem to be very different between the various software packages. Okay? Right, so there's a good performance of most of those. One thing that is very important is that, as I mentioned, they are not oracle, and you don't really want the black box here, having a model and you don't understand how it works. Okay, animals are a bit like that, intelligence. Artificial intelligence too, but for mathematical models, you want to see the equation. So there are currently two answers to this question. The first is open source code. The other one is open science locked code. And there are advantages and inconveniences to both. There is open source. You can see the code and check it. But, you know, the core code of complex model can be 10,000 of lines, extremely complex, okay? The code can be ununderstandable, depends how it is written. The software platform, not the code of the model, but all the engine to run the model, make prediction, integrate, whatever, those technical aspects, can be now millions of times. How, and how do you know what it really does? Okay, and the problem is that code and software, or there's a problem when code and software can be changed without control by anyone. I give you a code, I told you that I have done my simulation with that. How do you know? I may have changed the code. And in this case, also intellectual property is symbolic. Okay, you give away your intellectual property in a way. Okay, the code may be free. Okay, but now to run it requires a lot of expertise. Just understanding what the code does, what to put in, and how to understand what it's giving out. So you need specialists to run them. Okay? But when you do that, typically those specialists, they have to make a living, and you pay consulting fees. Okay. On the other hand, with the open science lock code, which is what the company I work with is using their modus operandi, or their business model, if you want. You cannot see the code and check it directly, but the model equations are published in scientific literature, peer-reviewed. You can use them, actually, you can rebuild them and check that the software does the same calculation. The code and software cannot be changed without control, and they are stable. That's a big advantage for agencies like FDA. And intellectual property is preserved. The internal quality assurance is usually organized, well organized, because we have audits. We have people asking to see, oh, where are the documents that show that you have written the equation here and that you have implemented them? We have audits all the time, just to make sure. And the software costs money, but you get support, maintenance, and guarantees of work. So that's part about the code, but I wanted to just mention briefly, there's not only the code that is important, but those models have many parameters. The good news is that they are mechanistic models, and a lot of these parameters actually come from verifiable data sources. Okay. I have a pointer, but I cannot move it.
Can we get to the conclusion slowly, Frédéric, perhaps?
Yes, quickly. Okay.
Thanks.
Issues with robustness, and I said, yeah, I know I have few slides, but I talk a lot. Issues with robustness and assessment of complex models. One point is that what has been noticed is that when you have the open source model, many of them, and people are complaining about that in systems biology, you find that there was a study on this, our model directly reproducible, half of them you cannot reproduce the results. They don't work, for example. So that's a problem when you're a regulatory agency and somebody gives you a model that doesn't work. Jean-Loup seems to, and EFSA seems to have found a way around that, but that is still a question. Now, coming to public discussion of model, I think that, as I mentioned, this meeting is a good example of a useful discussion. I wanted to say that mathematical models are just a technology, are powerful, but they are not good or bad by themselves. Okay, and have not been created to really obfuscate the environmental issues. They can have been sometimes used for that, but we should be careful to avoid it. Everybody knows that. Most of these models can only be run for computer. Okay, so the code needs to be safe, reliable, documented. And I don't think that there's a war to have between open source and commercial software. Open source is not usually completely free. There are hidden costs. Commercial software is often free for non-profit organizations. And I think that commercial and open source software are complementary. Commercial software is usually more reliable, and for people who don't want to develop, they don't even want to see the equilibrium. They can see them in publication, but how many people will really want to spend their time looking at equations and writing them? While the open source code will allow you to tweak your model, and it's more research tool, so they are not used for the same purpose in the same way. Okay, and there's a difference between them. Okay, so I think also that those models are very complex and hard to understand for the non-specialists, and it is essential that specialists from different point of view, industry, public bodies, but also association. I know some people who work in NGOs who have a very formal training in this type and understand the issues in those models, so that they can access the underlying science through peer-reviewed publications. And that's my conclusion on those various points which are open for discussion. Thank you very much.
Thank you very much, Frédéric. You really went to the heart of those issues, and I think, yeah, you are right in a sense to recall what's the right benchmark in terms of transparency, where we're coming from and where we're going, sort of, and you're right to recall, which is also what I meant to do initially, that we never had really full transparency on risk assessment, because there are various black boxes involved, the rat, the brain of the risk assessor or hazard assessor, and others, and in a sense the codes and the mathematical equations and the data are indeed perhaps more accessible than other black boxes, which is perhaps why we're putting the demands in terms of transparency perhaps higher there. There's a question here from the audience: is there a way to popularize or communicate more efficiently on what is in the black box to increase confidence in the results from their models? Perhaps that, I don't have more detail, but perhaps that speaks to perhaps the more complex modeling techniques you've alluded to, based on the artificial intelligence, machine learning models. We've seen that.
Yes. I mean, we well, first, for example, we are involved in some project, European project in this case, where we have public meetings, in particular with regulators, but we invite people also from NGOs and explain what we are doing. Okay, so obviously in those projects we develop new in vitro models, etc., but also mathematical models, and we try to explain as simply as possible, at least, what is the gist of it, what is at the core, what are they supposed to do, and not go into technical details. So I think there is space for more explanation to everybody about those. This would be difficult. I think that what would be interesting would be to explain really to people what and how artificial intelligence models work. Because in a way, they are not so much black box. It's a bit more subtle than that. We could explain to you, and in a way that almost everybody would understand if presented well, how they work. What is more subtle is that the results they give depends on the training of those methods, and this is where there is a kind of intelligence, if you want, which are very complex model which nobody understands. So if you want, the principle is quite simple, but the result and the way they work is not so simple. But at least you could understand what is happening. And I think it is worth explaining all that in conferences like this, or science, citizen science meetings. Yeah.
Mm-hmm. Yeah, very interesting. So there is hope in a sense. I mean, as you presented on PPBK, over time, over 20 years, sort of the places and also the community of specialists that is available to check models, verify models, explain models has been put in place. So perhaps time is needed for this to happen, for new kind of modeling techniques based on artificial intelligence. So okay, well, thank you.
We have to be all careful. Thank you.
Exactly. Thanks once more. We'll move to the last presentation of the panel, and so I'm happy to give the floor to Anax Oliveira. The floor is yours, Anax. We cannot hear you, Anax, for the moment. Or I cannot, at least.
Can you hear me now?
Yes, yes.
Okay, okay. So I'll leave my camera on to see how it behaves, or if I need to switch it off. If you're having connection issues, please just let me know. So thank you to the organizers for having me today at the session. I think it's a very important panel for us to be part of, and thank you all the attendees in attendance. I'm going to share a little bit of perspective about what we've been doing in terms of applying in silico tools to risk assessment framework as part of a regulatory setting, and then share a little bit about our perspectives for the future when it comes to what we are planning to develop as next tools for this specific application. So I think the discussion from the previous presenters were all very helpful to set the theme for me, and hopefully this is going to be easier now. Just to remind, and I think a couple of the previous speakers already talked about this, especially Frédéric, but I think it's important for us to have in mind that when you talk about in silico models, there are several types of potential applications, and each of them has their own weaknesses and potential, and they should be used properly in order to assess specific questions or problems. I'm going to focus today on the example that I'm bringing on the QSAR and machine learning tools, but it's important to be aware that those are specific tools within this world of in silico solutions for toxicity assessment. We are all aware of some of the gaps and challenges that we have to face in this space, and most of them have had pop up today, so I'm not going to be too much focused on this. But I just wanted to highlight two important things. We need to be aware that most of the in silico tools that we currently have available are relying on animal models. And this is because, of course, this is the type of data that we have. We historically have been built our risk assessment based on animal data. So that's what we have available now to actually create models, which means that the output of those models are actually susceptible to this weakness in terms of what is the human relevance, for example. I'm not going to talk about the black boxes and the difficulties to interpret because it was already very well discussed by Frédéric. But we should also bear in mind that every day we are facing more the challenge of modeling for complex endpoints, like for systemic, non-reversible endpoints, for example. In some cases, for things that we don't have clarity about what's the precise mode of action, which makes creating models even harder. I wanted to also echo one of Frédéric's points in terms of it seems that people have huge expectations for the in silico models. Somehow they should overcome the performance of animal models or in vitro models, for example. And I think we should bear in mind that these are only another different type of approach and different type of tool. It has its limitations, it has its potential, so we should acknowledge it rather than expecting that we be 100% transparent, comprehensive, and all of those other expectations that we know. And lastly, I wanted to touch base on the challenge of balancing appropriately between the quality versus the quantity of data in a time where we are all facing what everyone calls about of big data era. In terms of the applications, I like to always to make the distinction because we know that in silico tools can be applicable in what I'm calling here as exploratory space. But most of them, if we talk about risk assessment, we should expect that they are actually prone to be used as part of regulatory submissions, for example. So if you think of the exploratory space, I think that we know that in silico tools can be used from screening, prioritization, decision-making. We had examples of this already in Kate's talk today. So essentially, they are guiding the development, and the expectations that we have are different from what we expect to see in the late stages of development when we expect to have submissions made using those results, for example. So in the regulatory space, I just wanted to highlight we have, of course, the OECD guideline talking about what are the expectations for in silico models, but also we have mentions to the application of in silico solutions in different types of regulations for different types of industries, so for industrial chemicals, cosmetics, drugs, and medical devices, for example. I don't know what happened to my slides. I'm going to be focused now on a practical example of the ICHM7 to show a little bit where we are in terms of the application of those tools now. So the ICHM7, I don't know if you are familiar with this, but this is a guideline that is focused on the assessment of potentially mutagenic impurities that are contaminants of drug products, so it's applicable to the pharmaceutical industry worldwide. It is essentially focused on those impurities that arise from the synthetic product of the drugs. I think this topic became very famous recently with the several cases of recalls from products essentially from containing high levels of nitrosamines in the last 2 years, and this is becoming a more visible thing to the whole scientific and global community. But what's important about this guideline for our discussion today is that, as far as I know, it's the first guideline that actually allows the replacement of an experiment by the combination of two different in silico approaches as part of our risk assessment. So the ICHM7 brings this table here where the applicants are expected to classify their impurities in essentially 5 different classes, depending on the level of information that they have for each of them. So the idea is that they are classified as mutagenic, carcinogens, or non-mutagenic, for example, and something in between these. What I wanted to highlight is actually the fact that, as I told you, the guidelines set the space for the combination of 2 different QSAR methods, And those methods should be complementary. So they expect that one of the methodologies is expert rule-based methodology, and the other one be statistical-based methodology. And another important layer of this is that, sorry, I don't know what's going on with my slides. One important piece of this is that the guideline states that we should combine the assessment of a human expert of those predictions to know where we have sufficient evidence to actually make a final call, or we need some extra level of information to make a classification for a given chemical. Talk a little bit about the models. I'm not a developer of those models itself. I'm a toxicologist as a background. But what's important for us now is to understand what's the thinking behind each of those different approaches. So the expert rule-based system, it's essentially a knowledge base that contains structural alerts and reasoning rules. So what I expect to see as an output of this type of system is that we usually do to say in an easy way, is that I expect to see the result that a human would give for a given chemical if I ask what are the toxicological endpoints that could occur. So the system is basically looking for structural alerts in the database and rules to actually say what's the potential of this specific event occurring. And as a result, I would expect to see, for example, in the ICHM7 use case, that my compound is mutagenic, for example, given to the presence of this specific epoxide alert here. This is just to show a screenshot of how this type of system looks like, because essentially it relies on human knowledge and literature data. So you are expected to see what are the reasons why a specific alert is fired for your compound, but also what is the evidence supporting this specific conclusion, and some literature data to support this type of prediction. Whereas if you think about statistical systems, they are more likely to be we are expected to see what is the response that a machine would give if I am asking this type of model, what are the structural fragments that are in this domain, and is my compound mutagenic or not. There could be several ways to approach this. The way that we approach it at LHASA is that we are actually using fragments and seeing how many times those fragments are associated with positive or negative responses in the Ames test, for example. And we have a very clear way of calculating what is the level of confidence that I have in this level, in this specific prediction. So in the end, what I expect to see as an output of this type of system is a positive or negative response with a level of confidence associated with it. And this is, again, just to show you the output of this type of system. So here, the user is actually able to know where the prediction is based on, what are the most similar chemicals from the literature that were used and selected to actually give my result. And the user is also able to actually play around those compounds and see what is the level of information that we have for different strains that were tested in the presence and absence of metabolic activation, etc. So in the end, if you can combine these two different pieces of approaches, what we expect to answer is what are the bases for this specific prediction? What are the evidences that you can show me to support the hypotheses that were generated? And how much detail do you have about this evidence, for example? So what we do here in the end is actually increase the level of transparency and the volume of supporting information that we can provide to the user for actually he or her be able to, he or she be able to actually interpret the results and make a final call as the guideline expects. And as I've mentioned, the expert review is a very important layer of this process, and there are a lot of publications in the literature showing how actually the combination of the two approaches with an expert review increases significantly the predictivity and the accuracy and of this type of prediction, this type of approach. As you can expect, if you are using two different models, we don't expect that they always agree, because it means that if they are always agreeing, they are not complementary. So that's when it's very important to have the expert knowledge to allow to understand why the systems are disagreeing and what should be my final call. Do I need to actually run an assay for this specific case or not? Now, this is what about the past, somehow the past and the present, so what we've been doing, and a good example of how we are applying silico tools as part of regulatory frameworks now to support this type of decision. But as we all know, we are expecting to approach a position where we start to have increased number of in silico tools being part of this type of risk assessment applications. And we start to see a reduction in the number of animal studies that need to be conducted. So for LHASA, one very important concept that we are working a lot on is the concept of AOPs, which is basically a way of organizing the mechanistic information in a very structured way. And the way that we are approaching this at LHASA is that we expect to move from discovering AOPs to actually be able to organize them in a standardized and agreed way across the industry and regulators, add some level of reasoning between the different key events to understand how much information I have for this specific key event and how this relates with my adverse outcome in the end, but also to combine some prediction models for the molecular initiating events and also some pharmacokinetics models, for example, at the end, to actually be able to move from hazard to risk predictions. As you can expect, the AOPs can be applicable across different stages of the chemical development process, so you would have different questions in different steps of the process, and this type of framework actually allows us to address all of those questions depending on the level of evidence and the stage of development that we are. Those being, for example, what is going to be the consequence of me hitting this target, which are the assays that could best predict this outcome for me, or what are the adverse outcomes that I should be worried about. And finally, if we are talking about moving from hazard to risk, what we expect to do is that we are able to actually add potency data for each of those assays that can predict each of those key events, and combine this with pharmacokinetics modeling, for example, for us to have a translation between in vitro and in vivo safe levels, so we can actually predict risk in the end. And at this last minutes, I just wanted to highlight one very important thing that LHASA is doing in this space, which is data sharing. So at LHASA, we have this mantra that shared knowledge means shared progress, and we have obvious reasons for that, and I think everyone acknowledges this. Our first experience started in the past when the members started to share some feedback for structural alerts in DEREC, and they moved to share some knowledge and then proprietary data on mutagenicity, and we could see a huge progress in our capacity to actually predict mutagenicity just based on the amount of data that were donated, and allowing us to be more comprehensive about the mechanisms behind this specific endpoint. And then in the future, this move to actually open the door for LHASA hosting a lot of different data sharing consortium in several topics. All of them are pre-competitive, and LHASA is always acting as an honest broker, so it's a trusted neutral partner in this process to actually host and share the data and control the access, but also to make develop useful tools out of this. Here are just some examples of the consortiums that are going on at LHASA so far, but what I really wanted to highlight is that, of course, everyone acknowledges the benefits of this, them being for example, allowing people to access high-quality proprietary data, supporting evidences for different types of applications, save time and money, reduce the rate of testing, standardize methodologies across different industries, and this is something that we can see very clear now, and of course, improve the types of models that we already have in place. So just some food for thought to conclude my brief presentation. We already know at this stage that, for example, the combination of two complementary silico approaches with an expert review has been proven to be effective as part of regulatory approaches. So the question that I pose here is, should we actually be able to use a similar approach in other areas for similar endpoints like mutagenicity, for example? Also, we already recognize at this stage that the engagement with the industry and regulators in early stages of the development of our tools is very relevant to make sure that we are developing transparent and useful things. One thing that we struggle a little bit is to understand what's the acceptable level of risk for each specific application that we are developing tools for. So we know this is more clear when you talk about cancer and genotoxicity, for example, but we don't have the same level of clarity for other endpoints. And I think this is very important from a developer perspective. And also, again, sharing data means that we are able to improve the potential to develop new solutions, but also to improve and keep developing the current solutions that we have available so far. With that, I would like to thank you very much for your attention. I'm happy to take any questions.
Thank you very much. Thank you very much, Anax. I hope you can hear me now.
Yeah.
Yes, thank you for a presentation like which, I mean, in a sense, similarly to Frédéric also shows, I mean, a sort of hopeful kind of process whereby expert systems and predictions can be, if not validated, at least, yeah, verified and considered valid and therefore accepted as an element of regulatory decisions on products. I mean, in a sense, I mean, you've just presented an example from the pharmaceutical area. What in your mind, in your view, explains that this level of validation and acceptance has been reached? I mean, I guess you were at the heart of that with LHASA and your data sharing system. Was that the key thing, or are there other elements that explain the progress towards shared verification and almost validation of predictive systems?
I think it is a combination of different things. So the first thing is, of course, we're talking about mutagenicity, which is something that we have a very comprehensive understanding of the mechanisms. We are talking about one endpoint for which we have a clear yes or no response, for example. So in the end, somehow it is an endpoint that it's easier to predict, even if you can have all of types of mechanisms by which a substance can be mutagenic. In the end, I think the Ames test is very well established. So we are departing from something that's very well established from an experimental perspective. So this allows us to build very strong evidence for the models that we are having. And also we have significant amount of data, for example, in this space. So this should be taken into account for this specific application. And the other thing I think is we are talking in this case about the safety assessment of low level of contaminants. I'm not talking about applying this, for example, to waive an Ames test for an active pharmaceutical ingredient. So I think we should acknowledge that we are talking about low exposure levels, which means already a lower risk, even though we know that some of those compounds can be very potent, for example, and still be active at very low levels. But I do think that this combination of very well established endpoint for which we have a huge knowledge, and also applying it for compounds for which we have lower levels of exposure, are the main triggers for this specific use case that I just showed.
Right, thank you. Yeah, thank you, Anax, for these precisions. Please do not hesitate to put questions in the chat to Anax. I mean, perhaps a follow-up question, which I mean to Anax, but that will that speaks to other presentations and what the points that other speakers made, is that it seems to me, and it's in a sense reassuring, but that there's a lot of I just say expectation management around prediction and computational tools, in the sense that over time the level of reliability that we can expect from models, the threshold of evidence, the level of evidence that we can again expect from a modeling exercise, the kind of objects you've just talked about, low-level contaminants. I mean, the kind of things on which it is reasonable to apply models and to trust models. I mean, all of that is being adjusted over time between modelers, risk assessors, perhaps even decision makers. So, I mean, does that speak to your experience? And again, do we have reasons to be I mean, hopeful in that regard, in terms of, in the sense that modelers are not sort of left on their own making predictions, but that they are part of a dialogue, I think, as Frédéric has put it, that helps adjusting again expectations on what we can predict and how reliably we can do so.
I can totally understand where this is coming from, because we are talking about a completely new thing. I mean, now it's not new as were like maybe 10 years ago, but still something new because it's not part of our daily work. before joining LHASA, I actually worked as a toxicologist in different sets of industries, so especially in the pharmaceutical, cosmetics, and personal care industries as a risk assessor. So I can tell you that we are, in the daily basis, we are using things that are part of our routine for a long time and that has been very well established across the years. Whereas now we need to move for something that's completely new and somehow people look at it as a black box. So I think there is a very important component of this process that is not only focusing our attention in actually developing models, but actually teaching people about how to use those models, what is the science behind this type of prediction, for example. I don't suspect, for example, I was thinking about, do we actually need to understand the code as a risk assessor to trust the model? But can we actually have access to another level of information that could build the trust of this specific model? So can I show the validation of this specific model against a specific training set, for example, to see, can I understand how the domain of applicability of this specific tool is defined for me to know if it is applicable to the type of chemical that I'm working with? So I think there are some other alternative ways of approaching this problem of the trust and the confidence in this type of model, and certainly that goes through very important educational piece, but also finding out what is the level of information that we should provide for people in order for them to trust. Because I don't think that sharing the codes, for example, is going to be a single solution, because in the end, as toxicologists, we know we work in a very multidisciplinary environment. And I have no expectations that, even working for a developer company now, I'm able to understand every single line of a code that people there, which are developing these tools, are working on. So that's how I think it, and I do think there is hope for the future. I do see a lot of initiatives in this area going on. So I think we are on the right direction.
Right. There's a follow-up question from the audience, from Annabelle Demy. I mean, what's the process to define, to identify the endpoint on which we can sort of reasonably consider we're going to be able to gain some level of predictability and develop also a model? What's the process there?
I think one of our key drivers is always what is the industry and the regulators' needs. So the ICH M7, for example, came into place, but before that there were a lot of discussions around this. So actually we could clearly understand what's the problem and look to see if we could address it. So I would say this is one of the main drivers. But also, of course, we are a research organization, so we keep looking at what are the other things that are out there, for example, that we could be good at predicting. So the process is more about, can we understand the problem as it is? What are the other endpoints, for example, that we know that based on the toxicological and the biology knowledge could fit and be modeled by some of the approaches that we have expertise on, for example, right now. And there is always, of course, a high dependence on how much data we have available for this specific endpoint. Are we actually able to model for this depending on the level of information that we have available, either from public sources or from proprietary data that LHASA has access to.
Right, thank you. Yeah, very interesting. Perhaps we're moving now to a more general discussion with perhaps the various speakers can come in. So yeah, great. We could stop the presentation sharing, and if the other speakers want to reopen their cameras so that we can all be part of the discussion. Great, we can take, I guess, a few more minutes for that discussion. We have a question here from the audience, a long one. Let me read it. The panelists have discussed the limits of showing the code and full transparency because decision makers and stakeholders have a limited understanding of the latter. To what extent would the provision, better understanding of the, to what extent would the provision of quality standards behind science to decision makers and the general public be a solution? I guess that's, I mean, from that question, the expression the general public is is interesting. I guess it speaks to one of your points, Frédéric, which is, I mean, you mentioned several times that you know of NGOs who look at models, consider models, even use them. There's a broader point, I guess, here in terms of how is it possible to enlarge the dialogue around models and those tools, and how far and what kind of actors can we include in that dialogue to extend sort of peer review and make those models and tools potentially more public tools with obvious limitations. I mean, it can't be fully open, obviously, because of the various competencies, literacies of people, because of well, for many reasons. But yes, how far can we enlarge that that dialogue, and what's your experience in there?
Well, I think there are two elements of response. One is in terms of broad operational actions being taken. OECD, so I know OECD is not your next door neighbor, but OECD, WHO have large international conferences and actually actions on writing guidelines and getting not only industry, not only the various regulatory agencies from various countries, but also academics in universities or public research labs, and also stakeholders, part of NGOs, involved into those who want, but at least they are invited. Try to diversify the stakeholders involved in those writing guidelines, discussing how the models should be communicated, validated. So that's the first thing that is happening, and it's important. The second is similar or is linked. It is important for every organization to try to invite this, people in NGOs who are competent and have manifested their interest in the subject matter. The competence doesn't need to be a very pointy technical competence in writing C++ code, but it can be a competence in understanding the biology and mostly also understanding what is at stake. And we have actually seen quite a few PhD students. I have seen quite a few PhD students who do not continue in academic, purely academic or pure research. There's nothing like pure research, but field, but move to more open or local government positions where they have a training, and we have trained them in this type of complex modeling and complex thinking, but now they are applying it to more day-to-day, or if you want, a more pragmatic question. So it is very important that we keep and think and be reminded to invite those people.
Mm-hmm. Jean-Loup, Anax, what's your experience, if any, in terms of interacting with those stakeholders too, and NGOs, and bring in some other sort of public interest groups and other actors to discuss data, models, predictions? Is that happening, and is there potential here?
Anna, you want to start, Anax?
Yeah, I can go if that's okay. We recently have been a couple of discussions going on at LHASA about the quality piece of things, for example. So what's the best way to actually document everything that we are doing? How can we guarantee auditability, for example, when that's required? But this from an internal perspective, and if you think about an external perspective, we've been partnering with a couple of NGOs as well to work towards more of the understanding and teaching people about how to interpret, how to use our models, etc. And also with some scientific societies like the British Toxicology Society, for example, to actually be part of educational programs in those aspects as well. And lastly, of course, as I told you, we always try to engage with regulators in every step of the development of our tools, so we can make sure that what we are actually showing to the user is going to be sufficient, because in the end, the regulators are using the tools as well to check submissions when they got those submitted by industry.
Thanks, Anax. Jean-Louis, then?
Yes, so, well, a couple of examples. There's currently quite a large effort ongoing at the OECD level on this guidance document for the development and validation of PBPK models. That's ongoing. I think the document ought to be published in 2021. Same as Anax mentioned also, at Eurotox, we've actually created a new specialty section called Into Tox, which is in vitro in silico toxicology. And this provides opportunities to further discuss models at Eurotox, so symposium dedicated to QSARs or PBPKs or PBPK-PD models. And I think that will help also education, as Fred mentioned. So, of course, during those symposium, all stakeholders are present, whether we're talking about industry, NGOs, academia, regulatory agencies, and so on. So I think further work in this area will help to disseminate and communicate and stimulate the scientific community to further develop those tools and produce case studies that are meaningful for risk assessors, which is often, it seems like we ought to also tier the tools from the very simple tools to the more sophisticated tools, because often they are reluctant to use the tools. So we have to be quite didactic and proactive in testing. For some questions, maybe simple tools are sufficient, and maybe for some other questions we need more sophisticated tools. So that's some meditation. I think we need to make further meditation on that. That would be my take.
Sure. I have to. So unfortunately, Kate Guyton cannot be with us. She's having connection problems, apparently from the other side of the Atlantic, from the US. So unfortunately, she can't be part of this final discussion, which is a pity. In fact, I mean, a footnote to the discussion about NGOs and stakeholders. I had invited Jennifer Mark-Partland from the Environmental Defense Fund over in the US, who has done a lot of work in monitoring the what was being developed by regulatory agencies and research bodies in the US in the context of the Tox21 program, and she would have a lot to say about all this. But it's again very interesting that these dialogues are happening. Perhaps one word again about these simple tools that you were talking about, Jean-Louis, and I mean, it seems to me that it's also a trend, but I want you to perhaps hear your view on this. I mean, to have a broader sort of data platforms and light generic models to run on these platforms. I mean, and rather than develop highly sophisticated and in PPBK multi-compartment tools that would be less easy to interpret and work with.
Yes. So.
But perhaps I'm being too optimistic. What's the trend here in terms of developing tools that are easily accessible for risk assessors, expert committees, perhaps not decision makers, but at least for some people who don't have high level of initial literacy in math and IT and
so there's a couple of things I ought to mention. So the first one, there is a current international effort called APCRA, Accelerating the Pace of Chemical Risk Assessment, which is a consortium with a large number of agencies. So we started this work in 2016 with US EPA, ECHA, EFSA, the Japanese agency, Singapore, and so on. And we are trying to develop case studies to illustrate the use of NAMs in risk assessment. So the HTTK has been used in one case study that is published. The first author is Katie Friedman. It's been published in Toxicological Sciences to see whether we can predict point of departure using in vitro data and reverse dosimetry. Maybe you've seen that. There's a new case studies about to start on the further use of HTTK and incorporation of kinetic information. Another thing I ought to mention is, of course, in EFSA, every time we publish a guidance document, we have public consultation, so all stakeholders are involved. And the pesticide panel published a couple of years ago a review on TKTD models for looking at TKTD in aquatic organisms. So there's a guidance document, there's actually a scientific opinion, but it seems like there are more efforts needed to actually bring the ideas of having from simple to more complex models in a tiered approach into guidance documents that are quite practical for risk assessors. I don't know if I answered your question, David, but I tried.
Yes, I think the tiered approach is the key thing here.
So Frédéric,
Anax, to make a point related to this?
I'm shaking. No, I have a point here. I think also that sometimes in academia, so I take an academic hat here, we tend to overcomplicate things, and maybe in other places too. I was involved in a project where we were looking at bacterial infection in drinking water and just estimating the probability of infection, and we had started doing something very complicated, and in the end I realized that just a very simple model, very simple, that you could work out in Excel, for example, was enough to give good prediction. And interestingly, we published that, and for 15-20 years nobody talked about it. And somebody at a few years ago at WHO, they read the paper and they found that it was a simple way to do what they wanted to do. And since then it has been cited and used quite a lot. And I was surprised, it's just, I was happy. But sometimes we do too complicated things, and we can really, for some specific application, try to be simpler.
And I think we have to highlight again what Frédéric highlighted in his presentation. Sometimes we need basic data gathering to actually calibrate those models for different organisms. And I think that's really overlooked. Sometimes we don't have the basic data to develop the models for a range of species. And I think that would be good investment. If we are looking at, let's say, the ecological risk assessment, we don't necessarily have the physiological parameters for species of ecological interest. So it's definitely important that we gather structured data to calibrate those models.
Mm-hmm. Good data first.
I think if I can comment on the... Oh, sorry, we have a small delay.
No, go ahead.
If I can comment on the Frédéric's point about the complexity that comes from academia, usually I have the same experience when we talk about this piece of the adverse outcome pathways, for example. So I think the AOP Wiki is a huge and very important initiative in this area. But if you navigate across most of the pathways that are there, they provide so many levels of details at different levels of biological complexity that at some point it's very hard for you to actually translate that information into something useful in a risk assessment framework. So it's kind of similar to what Kate was bringing when she was talking about the key characteristics for cancer, for example. They had just 24 and they had to come down to 10 to maybe make it practical. So that's how we are trying to approach this problem now with AUPs in terms of, okay, you've just described a very beautiful and complex biological pathway, but what can we take out from here that actually could make sense for toxicological risk assessment? What are the actual key things that we should be looking at to make, and that are actually related with the adverse outcome that we expect to see here? So I think this is one of the challenges that we have when translating sometimes literature knowledge or knowledge produced in academia, which is, again, I need to make it clear, it's very important, but this communication should happen in order for us to have more translational tools being built upon that.
Okay, thanks, Anax. I think what I gather from those discussions is that there's a quite a high level of almost self-restraint, almost on behalf of modelers and their ambition to actually predict complex things and to adjust possibilities through a dialogue and mutual understanding with decision makers, risk assessors, potentially other stakeholders, and develop tools that wrong or not wrong, at least can be used. And I heard a lot of. I was not expecting that, to be frank, but I heard a lot of positive things around that today. We're getting close to the end of the session. Perhaps Frédéric.
I just wanted to,
what will be the last word?
I just wanted to say that it's much, in a way, it's easier to make a complicated model or to complicate a model because you put everything, you put it together. It's easier to complicate a model than to simplify it. And once you have a complex model, simplifying the model is something we don't really know how to do correctly. There is still a lot of intuition in it. And we can do sensitivity analysis, this type of thing, but they usually don't translate. We know that the model is maybe too complicated, but it's not easy to translate that into now what to do to simplify it, and please simplify it. And maybe we should try harder to do that. There's another aspect is that, computers are so fast, model is complicated, still takes a second to compute. Spend a lot of time simplifying it, it would take a millionth of a second. What do you care? I mean, between a second and a millionth of a second, doesn't show much difference for you. So there's not always an incentive to simplify the model in terms of computational code.
Mm-hmm. Yeah.
So that's just.
Yes, and and as a French singer, if you allow me, just a couple of years ago said in a song, simplicity is complicated, and one needs to want to be simple and understood and communicable, and that's not always the case. I guess we have to wrap up here. With these online meetings, I can't really ask the audience to join me in thanking the panel. I will not produce much noise, much clapping, but I hope the audience enjoyed these presentations and discussions as much as I did. I think we went to the heart of the issue. So really, again, thank you, Anax, Frédéric, Jean-Loup, for this webinar. Thank you, Kate. Unfortunately, she can't hear us. And yes, I hope you enjoyed that breakout session from the broader colloquium on the credibility of scientific expertise. Thank you once more, and good.
Thanks.
Thank you, David.
Everyone. Bye. All the best. Thank you, David.
Some clapping here.